So in the previous video, we have already talked about the cell signaling, the basics of cell signaling, and we talked about the different kinds of receptors. Now, mostly when we talk about signaling, we are talking about those receptors which are present on the surface of the cells. So we are mostly dealing with those kinds of receptors, or not those receptors which are present within the cell. Okay. So in this, we are going to discuss three major cell surface receptors. Now the first one, that is the ion channel coupled receptors. Now here you can see that the ions, once these ions they come and they bind to the receptors which are embedded within the plasma membrane. These ions binding, they activate the receptors and they lead to the opening of the channels. So you can see the binding of these ions, it has led to opening of the channel. Now this type of receptors, they are mostly found in the excitable cells. For example, we have muscle cells or we have nerve cells. So mostly occurs in those cases. The second type we have, it is the G protein coupled receptors. Now, if we have a closer look, we have here this one, this is an inactive receptor. The same, another thing we'll see that it is lying embedded within the plasma membrane. Second thing is we have an inactive G protein. We'll talk about when we will see in the another video, we'll see that the GPCRs, these G proteins, they are actually trimeric. So they are made up of three units. Now you can see that one of the units, they are tethered to the surface, to the membrane, to the plasma membrane. The third component, it is the inactive enzyme. Now what happens once there is a signaling molecule? So consider here. So once the signaling molecule, it binds to this receptor, it leads to the activation of the G protein. Okay. And once the G protein is activated, it leads to the activation of this enzyme which was bound to the membrane. Now, important thing about G protein coupled receptors, that is, so this membrane you see here, it is a serpentine. So that means it transverses the plasma membrane 7 times. Okay. So that is how basically mostly we'll see in most of the signaling processes, we'll be talking about the GPCRs. So that is how basically the GPCRs work. As signal comes, the receptor, it leads to activation of G proteins. The activated receptor and G protein, they lead to the activation of enzyme. And the activated enzyme, they will then lead to the activation of effector proteins. Like we saw in the previous video, we talked about how that effector protein, it can lead to the alteration of the cell metabolism, it can lead to the alteration in the cytoskeleton chains, or it can also lead to the alteration in the gene expression. Now let's discuss about the third type. So we have the enzyme coupled receptors. Now consider here, we have a signaling molecule that may come in the form of a dimer. Now once this signaling molecule, it binds to the receptor, again the receptor, it is lying in the plasma membrane, but there is one thing about the enzyme coupled receptors, they are not serpentine receptors, but here you can see that they have the receptor, it is having 3 domains. So you see here, this is the extracellular domain because it's lying outside the cell. This is the transmembrane domain, and this one, it is the catalytic domain, or we can say this is the cytosolic domain. Now, either it may be in these receptors, either it may be a signaling molecule in the form of a dimer, or it may be simply a signaling molecule. Now, in both of these cases, it may lead to the activation of catalytic activity, which may be within the receptor, within the cytosolic domain of this receptor, or it may lead to the activation of an enzyme that may be associated with the receptor. So the property, it may not be with the receptor. Okay. So the property, it may be with a separate receptor, but ultimately this is associated with this receptor. So that is how the enzyme coupled receptors work. We'll talk about these receptors in detail ahead. Now, so in your first video, if you remember, we talked about how basically a signal, that is an extracellular signal from outside the cell, it leads to production of the intracellular signals, and ultimately it generates the secondary messengers. Now secondary messengers, so here the secondary messengers, they may be either, so for example, they may be cyclic AMPs, they may be calcium. So these are water soluble, so they will diffuse in the cytosol, where some others, they may be, for example, DAGs, these are lipid soluble, and they will diffuse in the plane of the plasma membrane. Okay, so these are the different types of the secondary messengers which will be generated after the signal from outside basically leads to activation of intracellular signals, and that in turn leads to production of these secondary messengers, okay, which will have the same effect which we discussed earlier on. Let's here discuss about, now we know about these intracellular signals. So most of these intracellular signals, these are proteins which relay the signals by generating secondary messengers, which we saw just before. Now these proteins, they behave like the molecular switches. they will, so when they receive a signal, they'll turn on, and they turn off when there is no signal. So if you look here, so this is a signal. When the signal comes, a protein kinase. Now kinase, it is an enzyme which adds the phosphate group. Okay, so the phosphate group is added. You can see. So once the phosphate group is added, it leads to activation of this protein. Again, when there is no signal, now the protein phosphatase, it removes the phosphates here. Okay, and once the protein is removed, again the protein, it will go on into the off conformation, so that it is switched off. So that means this is the property is actually the phosphorylation, which basically mediates the conversion of these proteins from an on state to off state. Now, not only phosphorylation, now this signaling, it can be mediated by the GTP binding as well. Now, whenever this protein, it's bound to the molecule GTP, it is in the on state. Okay, so whenever there is a signal, the GTP, it binds to it, to the protein, it leads to its activation. But when the signal is withdrawn, the GTP hydrolysis occurs. So what happens is that when GTP is hydrolyzed, it will lead to the formation of the GDP and an inorganic phosphate. So you can see inorganic phosphate is released and a GDP. Now, whenever the protein, it is bound to a GDP molecule, you can see that now again it is in the inactive conformation. So two things we have to remember. One is that whenever it is phosphorylated, the protein, it is active. Whenever a GTP is basically bound to the protein, again it's active. And the opposite case is when there is no phosphate group, we say that the protein, it is in the inactive form, and when the protein is bound to a GDP, we say the protein, it's present in the inactive form. Okay, now basically these two processes, or the switching, it's mediated by certain regulatory proteins. So we may see here. Okay, so have a look. We talked about how if this is GTP bound, that means it is active. So you see this is the mark that the protein, it is now active. Okay, so this is an active monomeric GTPase. Now we have certain regulatory proteins. So we have GAPs. These are the GTPase activating proteins, and we have the guanine nucleotide exchange factors. Now what GAPs do? These gaps, always remember that these gaps, they lead to the inactivation of the protein. So they remove the phosphate group. For example, this is phosphorylated here. So yeah, for example, we say this GTP molecule. Okay, so whenever you have a regulatory protein, GAP, it's converted to the off state when it is bound to GDP. Okay, and the regulatory protein which basically does this, that is the gap. Okay, the second thing is the guanine nucleotide exchange factor. So when we talk about these GIFs, what they do is, so you see here we have an inactive monomeric enzyme. Okay, we have a protein which is inactive because it's bound to GDP. Now whenever there's an exchange, now the exchange occurs, the GDP is released, it's replaced by the GTP. Now again, when this is bound by the GTP, we see that it's in the active monomeric GTPase form. So that is basically how different types of the proteins, they basically regulate this switching between the inactive state and the active state of these enzymes. So this is basically how this type of switching mechanism works. And we'll see briefly in detail in the ahead videos how the different types of the signaling works in our cells.